Good afternoon, everybody. For those of you who I haven't met, my name is Mike Clagg. I'm Dean of the school, and I want to welcome you to this Dean's Lecture. And I want to thank Kate and Oren for trucking people in from all over the world to be here for this. I've never seen so many blue visitor tags at a Dean's Lecture before, so it's a great testimony to your drawing power. So we do these to recognize the accomplishments of faculty who have been either appointed or promoted as professor. And as I've said many times, it's difficult to get on the faculty at Johns Hopkins, but it's incredibly difficult to reach the rank of professor. And it's a decision that's made, it's suggested by the chair and the departmental committees. It's, I approve of it, but the decision is really made by your peers around the world and people who weigh in on your accomplishments and on your impact in the field. And so it's really an affirmation of your work by people whose opinion we all respect. So it's a big deal, and that's why we celebrate it. And I have to say that when Kate came up for professor, it's clear that she has won the respect of people around the world. So I'm just, everybody, I always introduce people who are doing the Dean's Lecture, but it's always ironic, as I say, because mainly most of the people in this room know the candidate much better than I do, but I'm going to do a little bit of introduction just to set the stage. And so Kate was promoted to the rank of professor in the Department of International Health, and she's really an internationally respected and recognized vaccine clinical trialist, epidemiologist, and pediatric ID physician. And her work, as you know, has been especially in the area of pneumococcus. She's focused on reducing health disparities here in the U.S. on American Indian reservations through her work with the Center for American Indian Health. And internationally, she has made major contributions working with her colleagues to expanding access to pneumococcal vaccine to infants and children. In addition to that work on the pneumococcus, she's also worked on the epidemiology and prevention of other respiratory and enteric pathogens, including RSV, influenza, rotavirus, and hepatitis A. And I knew Kate peripherally before I became dean, but I knew of her work in Haiti, with the ethylene glycol, outbreak. It's on an outbreak, it's a poisoning there because of the effect it had on the kidney and the international response to dialyze those children. So Kate did her undergraduate work in chemistry at the University of Toronto, and she followed that up with an MD at McGill University, and then came here to our great satisfaction to get an MPH. She also completed a residency in pediatrics and fellowship in pediatric ID across the street at Hopkins Hospital in the School of Medicine. Kate was at the CDC. That's where she did the work. I think in Haiti, when you did that work in Haiti, you were at the CDC, and then at the NIH, and then we were very fortunate to recruit her here. And one of her first projects was to conduct efficacy trials of pneumococcal vaccine among American Indians. So we're proud of all our faculty. We're especially proud of you, Kate, and the work you've done around the world. And this is where the view out your kitchen window now, I take it. So please join me in welcoming Kate O'Brien.
So I was saying to Mike as we were walking over to the auditorium, this is actually one of the more difficult talks to give because it's personal and because you are all my colleagues and my friends. And so it poses a certain challenge and a certain bar. So this talk is going to be, as I titled it, Mountains Beyond Mountains. I'm going to talk about vaccine clinical trials, but within a certain context, which is the work that I've been able to do and some of the places that the work has taken me. Some of you may be familiar with this title of the talk, Mountains Beyond Mountains, in part because of this fabulous book by Tracy Kidder, which if you haven't read, I would strongly recommend to you. And this book is about Paul Farmer and his work around the world to prevent and cure disease. And when you look at the tagline to the bottom of this book, it says, inspiring, disturbing, daring, and completely absorbing. And I hope that you will find the subject of vaccine clinical trials, all of these 4 things, and perhaps I'll give you some examples of why they are these things. So I do want to start in Haiti, and not because of the title of the book, which has a slightly different meaning for me. So my interest in vaccines actually began in Haiti, and there is a very common expression in Haitian Creole, which is Deyemun Gen Mun, and that does mean mountains beyond mountains. And it's a curious phrase because it has a number of different meanings, and probably every person has a different meaning for what this is. There is sort of the negative meaning, which is after every obstacle, there's simply another obstacle, and that may be the view of some people of what Haiti is like. But there are certainly much more optimistic meanings that really connotate things like when you reach a high peak, it's an opportunity to see a clear view of challenges ahead, or the notion that opportunities in front of you are frankly inexhaustible, just like one mountain beyond the next. And so if we actually go to Haiti, at least at the beginning of this talk, to help you locate where Haiti is, it's in the Caribbean, just a very short distance from Cuba, and it shares the island of Hispaniola with the Dominican Republic, and of course they have a very clear border between Haiti and the Dominican Republic, which is not just a bright line on a map, but is also a very bright line in terms of geography. Here Haiti is on the left and the Dominican Republic is on the right, Haiti being a very deforested and burdened country for a lot of political reasons. And I first went there after finishing pediatric residency here at Hopkins. I joined Andy Ruff and Neil Halsey, who took the great leap of faith and hired me to go down there to work on studies of HIV transmission using breast milk to infants. And this is the kind of setting that we worked in Cité Soleil, in a slum area just in Port-au-Prince in the capital city. And the conditions, of course, living there mean that for anybody who works on vaccines, you know that the kinds of prevention activities are absolutely essential because the conditions are ripe for disease, and treatment of disease is not going to be the most effective way of assuring health of people. And this was the pediatric ward that I worked in at St. Catherine's Hospital, a far distance and a far cry in terms of facilities from what I had just been trained to do at Johns Hopkins Hospital. And the kinds of things that I saw there, so many of them were vaccine preventable. So on the upper left-hand corner are twins, in fact, who came in, one of whom is a healthy, fat, robust baby, and the other is completely emaciated, and one of them had tuberculosis and the other one didn't. Measles, of course, with a child here with keratomalacia, neonatal tetanus, Hib meningitis. And so then the idea here from the clinical work that I was seeing was there was just so much pathology that was vaccine preventable. So my first lesson about mountains beyond mountains was that if you feel lost in a mountain, you probably are. That's certainly how I felt, and you should call for help. And so that's what I did. There was a time when I was there where in the clinic for following up kids who had HIV, we started seeing lots of kids with BCG lymphadenitis, and that's this big lump that you see under this child's arm. And sometimes BCG lymphadenitis can suppurate and start to ooze pus. And we were seeing lots of kids who were coming in like this, and certainly I had not seen this before, and I didn't know whether the rate that we were seeing was normal and what I should do about it. So in calling for help, I called Neil, and my memory of this is that the entirety of our conversation after I explained to him what I was seeing was him saying to me, "Kate, think rates." And then the phone went dead. And as a clinically trained pediatrician with no epidemiologic training, I had the expectation that this is what I was supposed to do, is sit there and think about rates. And it felt more like this. Really, I realized I had absolutely no idea what he was telling me to do. But what eventually happened after much gnashing of teeth and wringing of hands We did succeed in doing an evaluation of this, and it did, in fact, get published, much to Neil's great astonishment. So we followed about 290 kids from birth to 15 months of age, and some of them were born to HIV-infected mothers and some of them weren't, and overall about 10% of these kids had a significant BCG complication. And what we found was that among the children who were infected with HIV, about 30% of them had such a complication, compared with only 10% of kids who were not infected with HIV. Obviously, a considerable relative risk, likely attributable to the fact that the child was HIV infected. But that was not the only thing going on. And so I did a little investigation of what was actually happening in terms of immunizing kids with BCG vaccine, and discovered that because of a misinterpretation of the WHO recommendations and because of a problem with the syringes that had been donated to this poor hospital, kids were getting about 2.5 times the recommended dose of BCG. And when we corrected that very simple problem, there were no further cases of BCG complications in these kids. So that was my first experience, not with the clinical trial, but with the power of vaccines, both the positive power and the unintended consequences of misaligning vaccines. So my second lesson about mountains beyond mountains and climbing mountains is that if you just keep walking, you will actually end up somewhere. So just put one foot in front of the other. And that's what I did after leaving Haiti and going to the Centers for Disease Control, which those of you who have worked at the Centers for Disease Control will never recognize this picture, because this is not at all what it looked like when Oren and Antti and others in this room we were at CDC together. And it kind of looked like this, although this is a little bit older picture. But this is, in fact, the building that we worked in. And you can see, of course, CDC was dedicated to vaccines has been since its inception, and this was also a time when I worked on vaccines that the steps forward led me to the Navajo reservation and coming back to Johns Hopkins to join Matthew Santosham at the Center for American Indian Health to really tackle some of the disproportionate disease burden that was being seen among children on these reservations. And in particular, this is for those of you not familiar with where the Navajo and White Mountain Apache reservations are. They're located in the southwest of the United States. And these are sites where we have field sites where we conduct vaccine clinical trials. And most of my work has been on the pneumococcus, which is a bacterium surrounded by a polysaccharide capsule with over 93 serotypes, but only a limited number of these actually cause the majority of disease. And this pathogen is carried in the nasopharynx and causes both invasive pneumococcal disease, sepsis, meningitis, and bacteremic pneumonia, but also causes mucosal infections like otitis media and pneumonia that is not bacteremic in nature. And the importance of the colonization, of course, is insofar as it transmits the pathogen to other people in the community and has a large role to play in antibiotic resistance. And so our interest in pneumococcus and in its prevention is really centered in this figure, which shows the rates of invasive pneumococcal disease are disproportionately high among American Indian, Alaskan Native young children compared with the general U.S. population. And if we just focus on the rates of disease among the Apache and Navajo before pneumococcal vaccine was in routine use, and compare that to the general U.S. population, we can see that they had rates of disease that were 3 to 10-fold higher than children otherwise in the U.S. population. And so we undertook an efficacy trial to try to evaluate whether or not a new vaccine, the pneumococcal conjugate vaccine, would be efficacious. And just focusing on the kids who were enrolled between 2 and 6 months of age who received 4 doses of vaccine, the efficacy trial demonstrated that the product was highly efficacious against invasive disease. So that, of course, was a major vaccine clinical trial, and our work has continued since then to follow the epidemiology of disease after the product was routinely introduced into the population. And so here we just have rates of invasive pneumococcal disease in Navajo young children over different periods of time. First, the period of time during the trial, showing children under 2 years of age in green, those 2 to 5 years of age in orange, and those less than 5 years of age in red. And then once the vaccine was in routine use, you can see the rates of disease dramatically going down. And now that we have an extended spectrum vaccine, we really have managed to reduce the rates to an incredibly low level compared to what was normative for these kids even 15 years ago. The differential in rates of disease remains compared to the general U.S. population because, of course, these vaccines are also used in the general U.S. population, and we continue our work to try and eliminate that differential. So putting one foot in front of the other led us to also look at what was going on in the nasopharynx because it is the source of the pathogen for disease throughout the body. And without going through the number of nasopharyngeal studies that we've done, I was thinking about the number of people who we've actually sampled in the nose, and these are the children in the community, over 7,000 kids that we've followed in studies of NP colonization, over 6,000 adults, and through this period of time, we've collected a sort of horrific number of NP specimens, over 23,000 NP specimens, which we've studied, to try and answer a whole variety of questions around the ecology of the pneumococcus and how it behaves in a community where we've undertaken this remarkable human experiment of inducing antibody in a way that has never been done before. And so we know that when you interrupt transmission of the pneumococcus in children because of vaccination, that has a huge impact on all of the people that those children encounter, both children younger than the age at which they can be immunized, as well as their parents and the other adults, young and old adults that they encounter in their daily life. And so we've, as we just continue to look at the next logical question, we've addressed a variety of things about NP carriage and how conjugate vaccine affects carriage, what the immunology of carriage, both natural and vaccine-induced is, as well as the invasiveness of the strains and the molecular characterization of those strains. We also wanted to know how long it takes to actually get indirect effects from introducing a vaccine into the community, and what the sequence of events are that lead to serotype replacement in a community, as well as these dynamics of transmission within households, and fortuitously the characterization of new serotypes that have been identified and have proliferated within the community. And so putting one step in front of the other also sometimes leads to interesting encounters, meeting people like Donna Shalala, the governor, Jeffrey Sachs, and most recently, really just so proud that the center was recognized for the work with American Indians and pneumococcus with an award that was delivered by Francis Collins on the left from the director of NIH, Kathleen Sebelius, the Secretary of Health and Human Services, and John Holdren, the White House Senior Advisor for Science and Technology. And this was a presidential award, and so a huge honor for the center to be recognized for this and to be able to represent the center and accept this award. The third lesson that I learned about Mountains Beyond Mountains is that when you get to the top of a mountain, it's a good idea to stop and look around because there are likely other mountains to be climbed, and you can have a pretty good view of them. And so even though we were focusing our work on clinical trials on the reservation, if we looked around at what was happening in the rest of the world, there were many vaccine trials of pneumococcal vaccine going on throughout the world. And you can see the distribution of where these clinical trials were taking place, very widely distributed and highly representative of different areas of the world. The question was, how could we compare results across those trials? And each of them had a certain reason why it was done. The trial in Northern California was the pivotal licensure trial, representing a general U.S. population. By contrast, our trial, which supported the licensure, was in a high-risk U.S. population. The trial in South Africa, it's a real pleasure and great honor that Shabir Mahdi is here, who was the primary player in this trial, was done in Soweto, and really the focus of this trial was in a high disease burden setting, which was highly represented by HIV-infected children, and so the question was, would the vaccine work in those children? And finally, the Gambia trial, and again, Steve Howie is here from the Gambia, a very important trial to look at the ability of this vaccine to be efficacious in a malarious area and a high disease burden area where mortality could be measured. And so putting these trials all together, over 83,000 children were studied in these vaccine trials, and the summary measure of vaccine efficacy against vaccine serotype disease was about 80% across this remarkable diversity of epidemiologic settings and children with different risk factors for disease. Now, invasive pneumococcal disease, although important, was not the only syndrome that was of concern. In fact, pneumonia is the most dominant syndrome for pneumococcus in terms of mortality. And so looking at both the pneumonia outcomes across trials and the NP colonization outcomes across trials was a very big challenge. And one of the things that we directed our energy towards and thought was really important is that we should assure that learnings across these trials could be applicable to sites beyond where the trials were done. And in order to understand where the differences in the performance of the vaccine were, we had to assure that the methods that we were using across all of these trials were harmonious so that differences could be attributable to the epidemiology or to the vaccine themselves and not attributable to the methods that were used. And so we worked a lot on standardizing with WHO the methods for how we do NP colonization studies, and this has formed the basis for a standardized methodology that has been applied both to the clinical trials and to observational studies for the past decade. And we standardized how we interpret chest x-rays, which are the foundation for understanding the impact of pneumococcal vaccine on pneumonia. So I think these were really helpful efforts within the community to leverage the experience across a number of different vaccine clinical trialists and harmonize what we were doing. The other way that we sort of looked around and looked at what mountains were ahead of us was this great opportunity to extend the vision of the work to children in the least developed countries and the opportunity for them to benefit from the vaccine. And that was the launch of the PneumoADIP that was led by Oren Levine. And the vision of that project was to improve child health and survival by accelerating the evaluation and access to life-saving pneumococcal vaccines for the world's children. And this was a remarkable project that brought together not only vaccine clinical trialists and epidemiologists, but also health economists and communications people to have a multidisciplinary team to try to influence the policy setting for pneumococcal conjugate vaccine on a global setting. Two of the big stumbling blocks that we had were the understanding of the disease burden of pneumococcus throughout the world. And so we undertook a study to estimate in the most rigorous way possible the burden of pneumococcal disease among children younger than 5, both the cases, the deaths, and the cases and deaths averted, specifically at a country level, which had not been done before. And so the results of that are shown here in this map. This is, it's always remarkable to show one slide that summarizes 3 or 4 years worth of work, but that's in fact what this is. So we did estimate on a country level the number of deaths among children, with a total being about 826,000 deaths every year from pneumococcal disease. And you can see in rates of mortality really where the focus of disease globally is, and that is in sub-Saharan Africa and South Asia. The question is, what drives pneumococcal deaths? Is it just big populations, or is it pneumococcal mortality? And on this slide, what we tried to convey is among the 10 countries that contributed the most pneumococcal deaths, what was it? Was it the death rate, or was it simply the size of the population? So every bubble here is the number of pneumococcal deaths, and on the x-axis is the rate of death per 100,000, and on the y-axis is the number of children under 5 years of age in the country. So as you look at bubbles going from bottom to top, those are countries that are bigger in size, and that may be the reason why their number of deaths is greater. And going from left to right, those are countries that have an ever-increasing rate of pneumococcal death. Now, remember that these are only the top 10 countries. There are many, many countries that have a lower rate of death that are not shown on this slide. But to just put this in a little bit of context, if you imagine the UK, which has a population size of young children about the same as Afghanistan, if the UK had a rate of pneumococcal mortality that was the same as Afghanistan, it would show out here on this figure. But that's not the rate of pneumococcal mortality. In fact, the UK has a very low rate, and therefore the bubble size and its placement on this figure is down here. So I think this emphasizes that we can really use these kinds of information to focus our attention on where interventions are most important globally. Now, importantly, there was not one clinical trial that was done in any of these settings, and yet it doesn't impede us. We can still leverage the knowledge of the vaccine clinical trials to apply policy decisions. However, a second barrier was understanding whether or not the vaccines that had been formulated were relevant for developing world settings. And so we undertook a different study to evaluate the serotypes that were causing disease in young children throughout the world, and trying to articulate whether or not there were significant geographic differences in the serotypes that would cause disease. What's shown in this figure then is the proportion of invasive pneumococcal disease attributable to serotypes in the 3 formulations of vaccine that were available or coming onto the market by region of the world. And what we can see here is that for the 7-valent product, in every region of the world, about 50% or more of disease was accounted for by the serotypes in that vaccine. And for the 2 products that are now available, the 10 and 13-valent, every region of the world has at least 70% of disease is attributable to serotypes that are in those products. And so the next extension was actually getting these vaccines out to the countries that most needed them. And I think this is really, a very celebratory figure. What this shows is the comparison of pneumococcal conjugate vaccine compared with HIV vaccine and the pace of vaccine rollout according to income groups of countries. So if we first just look at the red lines, what's on the x-axis is the number of years since the vaccine was first licensed, and what's on the y-axis is the proportion of countries that have a universal recommendation for the vaccine. So if we look at the low-income countries which have open boxes and the high-income countries which are in the solid diamonds for HIV, we can see that there was a 9-year lag between the time that the high-income countries reached 70% of the countries using vaccine and the same proportion for low-income countries. For pneumococcal vaccine, we're estimating that there's only a 2-year lag, essentially a 7-year escalation in terms of the rapidity with which these vaccines are being rolled out into the lowest income countries where most of the mortality is. So I think the success here is evident in this figure. And we estimate that over 450 million more infants will actually receive pneumococcal vaccine in the first 20 years of use than received HIV vaccine. So I think we're gaining traction on our movement of vaccine clinical trials into policy decisions. And so when we look globally at the introduction of conjugate vaccine, this is the setting in the world as of January 2012 with countries that have a universal recommendation shown in the darker green and those that are approved for introduction shown in the turquoise. If we only look at vaccine, the GAVI eligible countries, here shown in green, and project out to 2015, you can see that we're having an incredibly rapid rollout of pneumococcal conjugate vaccine into these poorest countries, such that 58 countries are estimated to be introducing by 2015, and 33 are expected to introduce by the end of this year. So the fourth lesson that I learned is that mountains can be unpredictable and sometimes dangerous. So we should be brave and flexible in our approach. I want to mention the very, very first clinical trial that I did, which was a study of pneumococcal conjugate vaccines in infants with sickle cell disease. And this was a phase 2 immunogenicity study. And I'm just highlighting here the three people, Jerry Winkelstein and Howard Letterman, were the PIs of this study, and I was brought on to essentially run the study. This was done between 1995 and 1997. It was a very small study. I have to tell you that of the sort of key people, the PIs and the key personnel, there wasn't one of us who had ever done a clinical trial. We didn't have an IDMC or a data monitoring committee. We had no statistician involved in this study. There were no HIPAA regulations. We had no SAE reporting criteria to the sponsor. We had nobody on the study who had any regulatory experience, and we had no staff who had any regulatory experience. We had a 2-page consent form, which I found last night on my computer after much gnashing of teeth and trying to find it. It was 2 whole pages. And we had 7 case report forms, which totaled 10 pages. And the reason I say be brave is that vaccine clinical trials have changed tremendously in the years that I've been doing them. And what I can share is that in the 7 trials that I've done, which are either phase 3 or large phase 2 trials, we've enrolled over 11,000 subjects in these 16 years. And comparing that very first trial to now, it's a must-have to have a PI who has clinical trial experience. You must have an IDMC. You must have a statistician. There are many HIPAA regulations, which are very complex. We have critical reporting timelines for SAEs to the sponsor and to FDA. For all of our trials, we have a full-time set of regulatory staff. Our most recent trial, it's about to launch, has a 14-page consent form compared to a 2-page consent form. We have 89 case report forms compared with 10 pages in that last trial. And so this is the difference. And I have to say that I think Barbara Walters said it best, which is women don't get older, they just get blonder. And so I think this issue of the regulatory burden that we have on clinical trials is something we have to be very brave about. In the HIB trial that the center did, I wasn't there at the time, but Bob Weatherholt shared with me that in that trial he was given a box, and that box was where he was supposed to put all of the regulatory documents for that trial, and it looked something like this. I want to contrast that with the regulatory documents that we had for the last trial that we just completed. These are actually 21 volumes of regulatory documents. So mountains and careers in academics and in vaccine clinical trials are unpredictable. And I'm not sure whether you think this looks like a little bit of a mountain range, but I certainly thought it did. But I'd like to reveal to you what this actually is. This is my number of publications by year, and it's unpredictable. So looking at that, I was looking at this and just sort of thinking, gosh, what happened during some of these periods? Did I just get stupid? But in fact, it was unpredictable because I think I was productive in a different way. In these 2 years, I had each of my kids. And in these 2 years, we were, as a family, posted to other countries, first to Trinidad and then to Switzerland for my husband's job. And so there are, I guess what I would like to share, especially with some of the students and some of the junior faculty, is that even a career at Hopkins has its ups and downs, and you can survive all of these things, and they're very rich experiences, and we should not shy away from them. And as we look to the future, and sort of being brave. I think we really have to recognize that our work and our jobs is changing dramatically. That the world is a very, very small place now. We all have a seat at the table, but the way that we work is really changing. And I want to talk a little bit about that. I do serve in a Department of International Health, and we do global health work. And I think it's not lost on anybody that the way that we function as faculty members and especially as clinical trialists and epidemiologists is very different and is going to continue to be different than how it has been for the past 10 to 15 years. The world is now part of our computers are part of the world, and unless we are successful in becoming collaborators instead of leaders and partners instead of having to control studies, I don't think that we're going to be able to have the same place and be able to serve the communities that we do in the way that we have until now. I think we have to change the way that we partner. And I think that means that we have to harness technology, and we have to be present in ways that we otherwise haven't. We have to be flexible. And we have tools at our disposal, but we have to make best use of these, and we have to be visionary and aspirational in how we do it. And that's not to say that all the hurdles have been jumped. I was reflecting on this timeline of vaccines and the vaccine landscape and the pace with which different products have come on the market and the pace with which infants around the world have been vaccinated. And of course, at the outset of development of vaccines and introduction of vaccines, very few infants, when taken on a global level, have been vaccinated. And as the WHO and the EPI program was introduced, that certainly expanded, and the number of antigens that we use has also expanded. But what we should be anticipating is that there's a huge pipeline of vaccines that is right in front of us. And so the work, although it will be different work, is not work that is ending. It's, in fact, work that is becoming more complicated. The way that clinical trials are done is more complicated. They're certainly more expensive. They're more detailed. And there's still plenty of hurdles to be jumped and challenges ahead of us. So my last lesson about mountains is you should always climb with other people, in large part because the journey is a lot more fun. And I really can't talk about this work without recognizing the large range of organizations that I've had the great fortune to be associated with, both here at the school and outside of the school, and the people. And really the people, it's always all about the people. So this is just a photo of the infectious disease group that I've had the great pleasure to lead over these years. And we have a lot of fun together. I'd particularly like to recognize Bob Weatherholtz and Laura Hammett, who has just joined us, and Larry Moulton, who has been our statistician for all of these trials over the years, as well as PhD students Angie Ike and Jean Millar, and especially my assistant Kristen Sheffield and our field director Kathleen Norton. We do have mountains out in the reservation. This is Ray Reed, who is a Navajo physician, has been on our faculty and part of the center since its inception. And although we don't only have mountains, this is Larry Moulton during the HIB trial, where sometimes the bottom of the mountain is also a dangerous place to be. And of course, support from Bob Black as my department chair, and then key colleagues, Oren and Ruth, without whom I would certainly not have nearly as much fun. And I'd just like to as well recognize people in organizations and collaborators, Anthony, who's here, and collaborators really around the world who have been phenomenal in making the success of the studies. And we do have a lot of fun. All of these photos, in fact, you may not believe this, but all of these photos have been taken from work-related events. And I would be happy to tell you some of those stories after at the reception. And of course, the greatest thanks goes to Matu, who unfortunately couldn't be here today, who has been my mentor ever since I was an intern and had moved to the United States from Canada and encountered him as my attending on clinical service. And so since 1988, Mathieu has been my mentor and supporter in all of these endeavors and all of these places that I've gone. So these are the 5 lessons that I learned about mountains, beyond mountains. And I think in following these, you can have great success and great fun in the journey. Of course, the purpose of the journey is really this, which is the assurance of health for young kids so that they have the opportunities that we have all had. And with that, I'm going to close and, in fact, tell you, Mike, that this is the photo from our house right now. Thank you.
So that's not East Baltimore, right?
This is not, it's part of East Baltimore, but yeah.
So
secret place in East Baltimore.
Thank you for a great talk. And, you mentioned the center receiving that award, but it was really you who was named as the presidential, I have the exact title. It was the Presidential Early Career Award for Scientists and Engineers, which identifies a leading young scientist in the U.S. And I should have also mentioned about the Sabin Young Investigator Award, the Vaccine Award, which is one of the most prestigious awards. Great talk, great talk. So how many of those mountain climbing pictures were they actually you?
Yes.
They were. Okay. Okay. So I'm going to ask. We taped these for posterity. So maybe I could ask Jim or Josh to use that pick up that microphone there and ask for people who want to make comments or questions to use the microphone. So Warren.
Okay, thanks. Great talk. I have 2 questions for you. The first question is, having seen the progression of clinical trials over the years, do you have 1 or 2 suggestions for ways that you think clinical trials could be streamlined without sacrificing patient safety? And then the second question, which I know is on the minds of all of us, is what's it like to meet the president?
Well, let me answer the first question first. I think from my perspective, so it's really just a personal perspective, I think the regulatory environment is not evidence-based, and I think that we have to challenge that. I think there are such tremendous regulatory and legalistic barriers now that don't actually serve the interests of the subjects. In fact, I think they're counterproductive, that I don't believe that the documentation of informed consent is either anymore because of the degree of complexity. And I think this is an area where, from an ethical perspective, and I think from an academic perspective, we could make some traction in trying to understand what informed consent really is and how to do it well. I think the other thing is that I think we need it may not be sort of how we conduct vaccine clinical trials, but I think we really have to also understand how communities understand research and what perception people have about their responsibility, that number one, they have benefited from all of the things that have been done in the past, and whether or not they feel a responsibility to contribute in the future. And so to me, unless there are people who sort of have that mindset of contribution, both for the benefit of themselves or their own child, but also for the benefit of the community, I think that's a sort of community level discussion that should be had. The question about meeting the president, all I can say is it was absolutely phenomenal. And I'd love to have a beer with him sometime.
So was that Larry Moulton or his younger brother? They look like, well, if I can just weigh in, because when I started at Hopkins, we had a 2-page consent form, and then when I became vice dean, by that time we had an 8-page boilerplate. And what drives it, you're right, it's not evidence-based, but it's really the General Counsel's office that drives it, because there's so much concern about being sued. And you know, that's evidence-based concern. I mean, institutions get sued all the time. So I'm not sure, you know, you said, you know, we can make some traction in that, but it is difficult because it's really driven by.
Well, Mike, if I can just respond to that, I think you are right. There is some boilerplate legalistic language that I think it's going to be very hard to unravel and move back on. But it's not only that. There is, from the perspective of the vaccine manufacturers, there is this compulsion now to disclose, feel as if you're doing the right thing by disclosing so much, every possible thing, every permutation combination, that it's not a meaningful set of content for a human being to absorb anymore. And it's similar to the drug product labeling. You can't read that and actually take that on as a lay person and really understand anything about a drug that, you know, that's a licensed drug that you're taking. So I think we perhaps have ticked the box, or we feel like we've ticked the box by delivering this huge set of content. But it's not actually meaningful communication.
But that's because you think informed consent should be to protect participants, right? Those 8-page forms are to protect institutions, not participants.
Yes, I think that's true. Yeah.
Any other comments, thoughts? No? So if not, we have some, I don't know if it's French wine or Swiss wine, but we have some wine and some refreshments outside where we can talk.
Great.
Okay, thank you. Great talk.
Thank you. Thanks for coming.